Okay, hi. Now I'm going to tell you a little bit about this cloning project for the HTR2B serotonin receptor. And I want Ali to do this because of a question we have about the JC polyomavirus. Okay, so JC polyomavirus is a small DNA virus and it affects about half the world population. There are 8 or maybe 9 genotypes, and you find different genotypes in different places. So where do these different genotypes come from? Well, one idea is that, the virus has co-evolved with us. So when modern humans left the African continent and went and colonized different places around the world, the virus diversified with us. Now that kind of works except for genotype 1. So genotype 1 of the JC polyomavirus is very different. And you only find it in Europe, Siberia, the Arctic regions. So where does this genotype 1 come from? So maybe it was transferred to modern humans from Neanderthals or Denisovans when our ancestors moved into Europe and met these, prehistoric humans. Now this is an interesting idea, but is it possible to test it experimentally? Because we don't have any Neanderthals walking around to try and infect them with genotype 1 polyomavirus, right? JC polyomavirus, right? So it's kind of difficult to imagine how we can do this. Okay, so this is where the HTR2B comes in, because this is one of the entry co-receptors used by JC virus to infect cells. And it just so happens that there are some small differences in the sequence of HTR2B between Homo sapiens, modern humans, Denisovans, and Neanderthals. So the idea is, what we want to do is to clone up the HTR2B gene, then mutate it to give it the Neanderthal or the Denisovan sequence, and then try and get ancestral genotype 1 virus and see whether it infects cells better when they express either the modern human, Denisovan, or Neanderthal HTR2B. So that's why we want to clone the HTR2B gene.
Hello everyone, it's Ali Bizri, and just before we actually get into the gist of this video, I'd like to go and run down the different experiments that we're going to be working on. First of all, we're going to be having a look at the restriction digestion, where we're going to be using specific restriction enzymes to cleave the fragments from their plasmids, and these fragments are the HTR2B and the vector mCherry2N1. That's number one. Number two, we're going to be migrating these fragments on a gel to check if we actually cleaved it correctly. Then we're going to be having a look at the impure gel electrophoresis results and after purification and seeing the difference between them. After that, we're going to be performing a ligation reaction to ligate the HTR2B inside the mCherry2N1 vector. Next, we are going to be transforming E. coli using these fragments. After that, we are going to be performing PCR to check the positive colonies that we got. After that, we're going to be seeing the results of a few experiments and we'll try to analyze them together. First of all, we're going to be extracting the plasmids from the positive colonies. Then we're going to be sending those extracted plasmids for sequencing to make sure that our fragment is actually inserted into the mCherry2N1 vector in the correct orientation. Then we're going to be seeing the results from the site-directed mutagenesis, and then we're going to be essentially just also going and checking the sequencing results after site-directed mutagenesis to see how we can distinguish between the mutations that are of interest and mutations that are not of interest. This concludes this quick intro into the rest of the video, and I hope you enjoy it. If you have any questions, feel free to stop me at any time during the presentation, and enjoy this video.